If it doesn't get cancelled, then I'll move, hopefully.
Good, good. So, is there time or you start bang on 11?
It's like we are going live on YouTube right now.
Ma'am, I
We are
request
already
you can check your slide is okay or
share.
presentable.
You
That is
can
what
just
I
share
wanted to do.
screen share. You can just check once.
Okay, just give me a minute.
No problem. Take your time, ma'am.
Screen share, right?
Yes.
Green color, bottom.
I know.
Shubha, that view
Yes.
is not coming today. That all of the panelists together
It's
in one
coming.
screen.
Okay.
It's coming. Yeah.
Mm.
Is it fine showing now?
Yeah.
Huh?
Yes.
Yes.
Okay.
Yes, ma'am.
Yeah.
It's so.
Okay.
Okay. So, Shoma, Neha, Jacinta. Jacinta, you have the script. Okay.
Yep, you want me to start now?
No, just give me 2 minutes. I want to set up the YouTube and.
Okay, no worries.
Can we stop the screen sharing now?
No, I just request. Yes, ma'am. Can we stop the screen sharing for 2 or 3 minutes because we need to announce? And okay.
Is it okay now?
Yes.
You
Yeah.
let me know
Yes.
when to share, all right?
Yes. Yes. Shoma, you asking for this screen share, this mode?
No, not this. I don't
Okay,
know.
then you can. You
Got it.
yes,
Gallery view.
Yes,
No, right.
yes, it is your.
Okay.
Ma'am, I need 2 minutes just to
I'll just-
share the YouTube and LinkedIn and Facebook.
That's just fine.
Okay. Okay.
So we are going live on YouTube now.
Yes, just I start the live in YouTube, but I need to spread this way.
You can just signal me when you are.
Yes, ma'am.
starting.
Just give me 2 minutes. Okay.
So, Jacinta, how are things there in your neighborhood and close to you?
Yeah, it's not bad. It's getting cold again.
Ah.
Business as usual.
It's so hot in India. I mean, at least in West Bengal, it's very hot.
So, ma'am, Shubhobrato and Soma are my MSc colleagues from GGU, Bilaspur.
Hello,
And
ma'am.
Shubho
Hi,
came
ma'am.
up with this idea. So I thought I should really join them. At least I will do something sitting at home with the baby.
It's
And,
a wonderful idea to get people across and listen, and also gives a we can have a staggered audience, which is normally not possible in a regular kind of an academic conference, because there you have people with limited interest coming in. So I like that part of the idea, but it can be very varied, but then it's very difficult because if you have varied audience, then you have to really scale down and speak in a little laymanish language. So, but an interesting concept.
but most of the audience here are like either PhD students
Yeah, I saw that.
or masters, and like all related to like either agriculture universities, or the audience is quite big, I would say, and it's very varied also.
So is it
So,
regular for the seminars that you organize to have so many participants?
Yeah, it's 19th webinar today, yours,
20th.
and 20th.
Yes.
Okay, 20th. So yeah, we have been asking, like we have asked you, so we keep our backup for the speakers for next 1 or 2 months at least. So still with this idea we are going, let's see what all things we
Yes.
can do more with the plant science.
All the
webinar.
best to you. A nice concept, and I'm sure you'll keep getting a lot of speakers.
Yeah.
Yes.
I'm really lucky that I can, be with you before I'm leaving. Otherwise, I was scaring like I will be in quarantine after my travel. So I was thinking I will miss your talk in between. Because when I asked you, nothing was on my plate at that time. So
because
I
it
was like,
so happens that there are so many talks now, given that you can be giving 2 talks per day sitting in your office, so the number of talks and the number of organizers has really shot up, and that causes all the stuff. But anyway, he gave me plenty of time, more than a month, so it was like, okay.
thank you so much, ma'am.
Yes, thank you,
I
ma'am.
honor.
So I want to start because it's time. Okay.
Yeah. I'll just get myself.
Okay. Hello, everyone. Today is our 20th webinar in this race. We are very glad that we have a Dr. Neeti ma'am for today's webinar. We have a small announcement regarding our new session. It's called Bioengine Discussion. It's dedicatedly for the master's, PhD, and early career scientists. We can discuss a little bit before other than that life science webinar. So I request Jacinta to introduce bioengineering. Then I will request Neha to introduce our speaker. Shoma will conduct the interview session, and I request all the speakers, all the listeners to keep your patience. At the end of this talk, we will give the link and password for today's certificate collections process. So I request Jacinta.
Thanks, Juvo. Welcome everyone to the BioEngine webinar series. This webinar series has been designed to build a platform from where plant scientists can present their research to the world and future scientists can gain perspective and inspiration. We are grateful that the webinars have been appreciated by our viewers from all around the world. We will soon start a new series, part of the BioEngine initiative, called the BioEngine Live Discussions. This is mostly a peer-to-peer discussion session dedicated to research in the field of life sciences. The discussions will include important aspects of our research life, such as research planning, conceptualizing, methodology, and project designing, as well as manuscript writing, finding appropriate journals for publication, excuse me, scientific communication, and national and international fellowships for PhD and postdoc research. You are all welcome to join this initiative and participate in these discussions. Please visit our website to know more and to register in the discussion session. You can post your ideas on topics that you want to be discussed in the live sessions. Our first live discussion is on October 17, 2020 at 12 p.m. IST or Indian Standard Time. For today's webinar, we have received a huge response with participants from 51 countries around the world. We welcome all the viewers and thank them for their interest in the BioEngine webinars. You can apply for a certificate of participation via the feedback link. The password will be provided in the YouTube chat box by the host after the presentation. Submission of the feedback form is mandatory to receive a certificate. Please be patient. You will get ample time to fill and submit your feedback form. The participation certificates can be collected from our website after 2 days. Please visit our website to register for upcoming webinars and live discussions. You can connect with BioEngine via Facebook, YouTube, Twitter, and LinkedIn. I now request Neha to please introduce our speaker for today's webinar. Thank you.
Thanks, Jacinta. So the topic for today's webinar is Mirrors to Solve Stress by Dr. Neeti Sanan-Mishra, Group Leader, International Center for Genetic Engineering and Biotechnology, New Delhi. Dr. Neeti Sanan-Mishra graduated from Delhi University with a bachelor's degree in Botany honors. She completed her M.Sc in Life Sciences from Jawaharlal Nehru University and went on to pursue her Ph.D. in the area of Molecular Plant Physiology from JNU. Thereafter, in 1998, she became research scientist at Center of Plant Molecular Biology, University of Delhi, South Campus. She joined ICGEB in the year 2000 and currently she is leading the Plant RNAi Biology Group. Her group's research interest is towards understanding the miRNA networks involved in regulating the plant development in response to virus infection, high temperature, and salt stress. The group is using bioinformatics along with the traditional molecular biology and biochemical approaches to understand the miRNA regulations operative during development and stress. She has several awards and patents to her credit. She is also on the editorial board of scientific journals. Dr. Neeti has published several research articles in the reputed international peer-reviewed journals such as PNAS, Scientific Reports, Frontiers in Plant Science, Plant Molecular Biology, and Virology. We are highly honored to have her with us today. I now request Dr. Mishra to please share her screen and begin her talk.
Good morning all, and for those who are in India, and thank you very much for having me on this seminar series. So is my screen visible now?
Yes.
Yes?
Yes.
Okay. So thank you all again for joining me to listen me on this topic, which is MirROR, that is microRNA regulation of response to salt stress in plants. And at the outset, I must thank the team BioEngine for organizing this talk, and all of you to join in to listen to my views on what a response of plants to salt stress is. And this is more so in the time of today's pandemic, when there's a lot of social distancing and we are all far from each other, and viruses have clouded our mind. Now, other than virus, the most important thing that has crossed everybody's mind is food, because as soon as the pandemic was officially acknowledged and a lockdown was initiated, people started thinking about food, about resources to keep themselves fed during this time. And also many of us were thinking about those who don't have the resources and how to feed those people. So underlined here is the requirement for food. And one of the steps to mitigate this hunger is by growing more food. And this is where the challenge lies in front of the scientists today, because when we talk about food, we have to produce it in a present environment where there is a lot of climate change, there is an expanding world population, and there is a lot of environmental degradation. So all this is producing a lot of stress on the plants, on the agriculture and the produce. Also, it is highlighting the importance of food for human welfare, because directly or indirectly it affects the human health. And when we talk about crop plants, within the crop plants, rice happens to be the most used cereal. It is not only a cereal, it has become a part of our life part of our rituals ever since it has been domesticated. It is no longer considered just as a food. It is very much a commodity, and is culturally a very important part of human existence. But what is the future of rice now? Oh, I have a screen which is missing. So what we need to understand is that rice is mainly grown in these Southeast Asian countries, and these are the ones which are facing a lot of climate change. And as a plant, paddy or the rice plant is considered most sensitive to soil stress during the early stages and the reproductive stages. And this directly impacts the grain yield production of rice. Now, the global climate change produces stress to the plants. So what do we mean by stress? Stress is when the ambient temperature or the ambient requirement for a plant to produce, to grow in a healthy atmosphere and produce seeds is disrupted. And this can happen if we have one of the environmental factors, which is either too much, comes in too soon or too late, the timing is changed, or is too less. So example, in case of water, which leads to drought. And all of these impact the growth and the productivity of the plant, which impacts is directly seen in case of the diminished quality and the reduced yield. Specifically, if we look at the higher temperatures, it has been seen that for every 1 degree rise in temperature, the yield is supposed to decline by is predicted to decline by about 7%. And the impact is even more when we talk about the nighttime temperatures. Well, this is another line of study, and probably I will mention this during one of the slides later in the talk. So, how does this impact rice? Because if we look at a country like India, then we can see that the rice plant is growing almost everywhere, right from the northern tip to the southern tip of the country. And there is rice which can be cultivated as an irrigated dry upland rice. There is low rain-fed lowland rice, which is mainly rain-fed. Those wet rice which is growing in the flooded regions, and there's also deep water rice which has the capacity to keep the heads above the rising water table. So the rice plant can grow in a variety of regimes. Yet I say that whenever there is an environmental fluctuation, it will impact the productivity of rice. So what we need to understand is that there is genetic information in the genome or organization of rice plant for making it able to adapt to grow in a particular niche, whether it is from overwatered to underwater regions. The capacity is there, but depending upon the niche or depending upon the area where it is growing, it needs, the plants have adapted themselves, and hence whenever there is a fluctuation, an impact is perceived. Yet there is ample potential in the plant to be able to grow in one of these changing environmental regions. And to understand this, a lot of efforts have been done by research groups all over the world. And if we look at the genome sequencing of rice, which started somewhere in the early 2000s and has progressed since then. And now we have more than 3,000, 4,000 genomes which have been sequenced, and a lot of information, different varieties are there. But why I am showing this timeline is that we need to understand that while this research had started in 2000 about sequencing the rice genomes, understanding or reading the genome, identifying what is written there, parallelly around the same time, in the 90s, began the revolutionary discovery of RNAi or RNA silencing. And 2002 was the year which has been acknowledged as the year of RNAi by science. So the two things ran almost in parallel with each other. And now, after almost 2 decades, we are at a position where we understand that the classical gene expression, along with that there is a silencing mechanism that is happening. So the information which is written in the genome is transcribed into the RNA and translated into the protein. But some of these RNAs have regulatory function, and then they can go and they act as switches to switch on or rather switch off gene expression. And this forms a important part of silencing. And it is not expression alone or silencing alone which is important, but a balance between the two, which will govern or which will fine tune the genetic machinery or the genetic program of a plant or of an organism, so that it can grow and thrive properly in its natural environment. And this area is still a black box. A lot of information has been obtained, and we are still on a path of discovery to understand what is silencing and how a balance or a dynamic interaction between this expression and silencing machinery is maintained inside the tiny cell, so that the optimum responses can be maintained. Now, if we look at this RNAs or the regulatory RNAs, they are a big family. Within this comes these non-coding RNAs, of which there have been many classes which have been recognized. And within this class lies the 20 to 25 nucleotide small regulatory RNAs. And one of the major class of these small regulatory RNAs are the microRNAs. And these microRNAs are the ones which are responsible for regulating the developmental phases of an organism. And if I talk in the terminology of plants, making us understand why a rice plant will look very different and grow very differently than, for example, another cereal, say maize or wheat plant. Now, for those who are new to silencing, I will just very briefly cover the basics of what microRNAs are. Like I said, they are endogenous. That means they are coded by the genes. So there are specific genes present in the chromosomes which will code for these microRNAs. They are transcribed as larger transcripts, which are these single-stranded molecules are heavily or compactly folded and are called the primary microRNAs. From one of the folds of the primary microRNAs, a hairpin-like structure is processed out by the action of specific proteins, mainly the main slicer being the dicer-like protein, or the DCL. And these hairpin structures are known as the pre-microRNAs. These pre-microRNAs are further processed to remove the duplex, the hairpin loop here, to form a duplex structure. And this microRNA star duplex is then methylated, and then all this process in case of plants occurs within the nucleus of the cell, and the mature microRNA is transported out into the cytoplasm, where it associates further with another complex of proteins to bring about the function, which in case of plants could either be cleavage of or a cutting of the target mRNA so that proteins cannot be formed, or these are translationally repressed so that the translation machinery is blocked at one of the processes and the proteins cannot be formed. Now, a lot of research and lot of exciting results have happened, and we know the other molecules, the helper proteins, the partners which are involved in this whole process. This is a highly regulated process. It may be sequence independent, but then there is a battery of proteins which are tightly regulated, and they will regulate how and when and why will a particular transcript be processed as a microRNA, and once it is released into the cytoplasm as a functional molecule, on what transcripts it will act, and how it will bring about the function. Now, this later, or the functional part, of course, is a sequence dependent process, whereas the processing happens to be a sequence independent process. So overall, generally, it is recognized that microRNAs repress the expression of target genes by either cleavage or translational repression, which form a part of the post-transcriptional gene silencing or the PTGS pathway. And there is another component which will go and switch off the transcription at the genetic level and will form a part of the TGS or the transcriptional gene silencing pathway. Okay. Now, just for people who are from the non-plant background, microRNAs are also found in animals, in insects, and all other eukaryotes. But there are similarities and differences in the pathway. And as a textbook knowledge or a basic differences between the two is, whereas the complete processing from the mature pride transcripts to the mature molecule occurs in the nucleus in the plant cells. In case of animals, it is the precursor molecule which is exported into the cytoplasm, is processed here, and then it gets associated with the functional protein complex to bring about mainly translation repression. MicroRNA mediated slicing is not known in case of animals or insect cells as of now, whereas both the mechanisms are present in plant cells. So this is just a very preliminary, a very basic textbook kind of knowledge to differentiate between the kind of the processing and the functional differences between the microRNAs. The molecules, however, remain the same. They are endogenous, and they can act on the endogenous transcripts. So when I say that microRNAs are produced from genes, we all understand that genes, there can be multiple copies, there are alleles, so on and so forth. And the same exists in case of microRNAs. There can be multiple copies of this of the MIR genes spread across different chromosomes, and hence different precursor molecules can give rise to many microRNA molecules. Now, there could be inherent or differences between these precursors at specific positions, but there are certain positions, specifically from position 1 to 10, which remain conserved, and there could be differences further downstream, it being a 24 nucleotide molecule. And all such sequences which have similar structures are normally grouped together and are considered as members of a particular family. Now, this is important, as I will come to it later, because normally the transcripts belong to large family members, and it is presumed that during evolution, the microRNAs have also evolved in such a way so that there are multiple members of the microRNA families to regulate multiple members of the transcript families, and we are still in the process of learning and understanding how this regulation and this coordination is brought about. We have some knowledge, but there is still a large amount of gray area which needs to be, the mist needs to be removed. Okay, so coming to how do microRNAs act, like I introduced that they can act through the PTGS or through the transcriptional gene silencing or the TGS pathway. The PTGS pathway will include mRNA destruction by cleavage, and the other would be the repression, which can occur through several means. And the transcriptional gene silencing is mainly by regulating the chromatin structure through methylation. And without getting into the details of the two processes, this is just to give a brief idea on how the different microRNAs act. So now, knowing that microRNAs are there and they can repress transcripts, the question becomes, what do they regulate and why do they regulate? Now, if we look at plants, the entire morphology of the above ground or the underground parts, which can be quite variable depending upon the plant species under study or under consideration, MicroRNAs can regulate almost every aspect of plant growth, whether it is development, we talk about right from seed formation, cell division, organ identity, polarity, the vascular development, and as the growth proceeds, the phase change, everything is under microRNA regulation. So far as students of science, we have studied a lot about hormones, the phytohormones specifically. So the hormonal responses themselves are regulated by these microRNAs. And other than that, they are also known to regulate the transposons, and this helps in maintaining the chromosomal or the genomic organization, as well as the stress responses in plants. And what is, just to show you the power of these molecules, or what the impact of these regulations are, this is what an normal Arabidopsis plant looks like. You tweak one microRNA and then it can change into a leafy mass. This is the capacity of a microRNA. You change one and you can change the entire phenotype of the plant, from a Arabidopsis plant to a heavily a vegetative structure, or you tweak another one and then the plant can change into multiple shoots or bolts, and will completely turn into a heavily flowering structure. So this is the impact of the molecule. It is a very, very powerful molecule, and it holds the capacity to change the phenotype and the physiology of the plant. So just to make you understand how the microRNAs act, knowing that each molecule is powerful and there are many of them. So I picked up this classical slide which talks about the development of C. elegans. Now in the insect, the life cycle can be divided into 4 broad stages: the L1, L2, the L3 or the L4 stage, and the adult stage. Now, if you look at the early larval stage, there is transcription of the Lin14 and the Lin28 transcripts, resulting in the formation of the respective proteins. And these respective proteins are required very early during the larval development. And as the larval stages progress, you see that there is the microRNA Lin4 will appear, and it will come and restrict the Lin14 and the Lin28 microRNA translation, so that the proteins are not formed. And this absence of the protein acts as a signal to push the insect from stage 1 to stage 2 or 3 or 4 of development. What you see in the lower panel is that during all 3 or the 4 stages, there is a transcript called Lin41, which is getting transcribed into a Lin41 protein. This protein acts as a repressor of another messenger RNA called Lin29. And this Lin29 message is constantly repressed in the larval stages. When the time comes to convert into an adult larval form, another microRNA, Let7, will appear. This Let7 will also go and keep ensuring that the 14 and 28 are tightly repressed, so that the proteins are not formed, because these proteins are required only in the early stages of the life cycle. And at the same time, they will also go and block the Lin41 mRNA. So the Lin41 protein is no longer produced. Now, since this protein is not there, the suppression activity is removed, and we get the synthesis of Lin29 protein. And this is how sequential appearance of the microRNA will regulate the gene expression, so that the developmental timing of the insect can be tightly controlled. So this has been very quickly and very conveniently expressed or explained through this cartoon, but it took a lot of hard work and a lot of mutant analysis to reach this kind of model, so that it can be easily explained to you people. Now, coming back to plants, similarly, in plants, we have a battery of microRNAs which keep coming and disappearing to intricately regulate gene expression. The classical example here can be the formation of a leaf, we all know that there is a SAM, or a shoot apical meristance, from which the leaf primordia are initiated, and then they grow and they form the young leaf, which then goes to form a mature leaf, and then senescence sets in. Now, each of these stages is intricately regulated by a series of microRNAs. Like in the initial stages, you have 3 of them which act on specific transcripts. And if you notice that most of these transcripts happen to be transcription factors. We all know that transcription factors are master molecules. They are regulating themselves, regulating the expression of hundreds of genes. So it is one microRNA regulating a particular transcription factors, and hence regulating the expression of hundreds of downstream genes. And the sequential appearance of these microRNAs will determine exactly when the leaf primordia will be defined, how it will grow, how the organ polarity will set in, so that you have an abaxial and an adaxial site getting determined, the young leaf getting formed, how and when it will mature, and when the senescence would set in. Now, among the different microRNAs here, I will take the example of one particular miRNA, which is MIR 156. And if you focus at the middle panel here, so this is how a normal Arabidopsis leaf would form. There is a young leaf which gradually matures, and then after a set time, the senescence will set in. And it has been observed, this is not time to time scale, but generally what happens is there is a microRNA 156. The level of this microRNA starts gradually decreasing. As the level starts decreasing, the level of its target transcript, which is SPL, starts increasing, and the SPL protein starts getting formed. So with the maturing of the leaf, the MIR level gradually comes down and the SPL level gradually goes up. Now, what happens if this microRNA is continuously overexpressed in the leaf? So you look at this panel now. When the microRNA is continuously overexpressed, there is a constant inhibition of the SPL transcription factors, and the leaves are found to remain in the young or the early stages. They do not mature. Whereas if you knock out 156, what would happen? Then the SPL protein will slowly start accumulating, and you can see that the lifespan of the leaf is drastically reduced. The leaf matures very fast, and also senescence will set in. So this is how, this is broadly to explain how the microRNA gradients will regulate the protein gradients in the cell to regulate the developmental timing in case of a plant. And and just to explain you, or to answer curious minds, once the SPL level increases, SPL happens to be an inhibitor of another microRNA, a promoter of another microRNA called 172. Now this, so the accumulation of SPL will start promoting the accumulation of MIR 172. MIR 172 will bring down the levels of AP2. And this AP2 is a suppressor of flowering. So as the AP2 level comes down, the signal for flower development will start, and a shoot apical meristem will slowly change into the flowering meristem. So these studies, there is a lot of knowledge, there is a lot of information available. It is very difficult to explain all of this in one slide, but the very idea about putting these 2 slides is to explain or to give an idea of how these microRNAs act. They act on target transcripts. In many cases, these happens to be transcription factors, and there is a gradient kind of effect which is seen in the plant, and this gradient will control the timings and the durations of the various phases in plants, insects, as well as other organisms. Now, like I said, that microRNAs are also acting on the hormonal pathways. So like you, here I'm trying to show a cartoon which shows an auxin signaling pathway. Auxin acts on its receptor TIR1, then downstream signaling pathways are activated, which activate number of downstream transcription factors, those mentioned in these yellow circles. And then these transcription factors will regulate downstream genes to control different aspects of plants like root formation, shoot morphology, flowering time, flower morphology, so on and so forth. Now, over and above this, you can see, look at the forming a layer, an integrated layer of regulators, and therefore these two regulators will dynamically interact with each other, and this will help the plants to continuously regulate or monitor their development in response to environmental cues. So so not only these microRNAs regulate the hormonal responses, their expression themselves are regulated by the auxins. So you can imagine the kind of complexity which occurs. So there are layers of we can talk about integrated layers of regulations which occur, and even the formation, a simple thing as a formation of leaf is not as genetically that simple. It is highly intricately regulated by a combination of transcripts and microRNAs to regulate the morphology, the shape, the time, and the various aspects of leaf formation. So before we move forward, it will not be wrong to say that when we talk about transcription factors, or when we talk about the small RNAs associated with RNA interference, they are both master regulators. So there are two master regulators which talk to each other. And why do I categorize them as master regulators? Because both are naturally occurring processes in plants. Both of them can impact more than one genes, and they regulate endogenous genes pathway. So if we can understand how these two talk to each other, interact with each other during the growth and cycle of a plant, because plants are sessile, they have to respond, they have to interact with the environment, they cannot run away from them. So an understanding of these two factors will throw light on understanding how the plant growth and development is modulated or regulated in response to the changing environment. So with this in mind, we have two aspects. Now, in one aspect, I'm telling you that microRNAs regulate development, and there is another aspect where I'm telling you that stress regulates the plant development, and both of them have an impact on yield. So there is a kind of a developmental plasticity which occurs, and this is because of interaction of the two master regulators with each others. So with this knowledge that alteration or abundance of microRNAs can lead to developmental abnormalities or disease state, we asked several simple questions before beginning our research into RNAi. The search was for the potential master regulators and to understand and how they function so that they can modulate the plant physiology and help them to adapt to stresses. And also whether, and during this process, we also took help from the viruses, because some of the plant viruses bring with them suppressors or inhibitors of these microRNAs and or the RNA silencing machinery, if I make it more generic, and they can be used as excellent probes to understand the function of the microRNAs in response to both biotic as well as abiotic responses to the plants. And of course, the ultimate question is, once we understand these microRNAs, can there be any such tools which can help to enhance the plant productivity, specifically in the present day where there are a lot of limitations and a lot of challenges before agriculture. So just to give you an overview of what my lab has been working on, we took rice as the major crop, although we are looking at other crops also like tomatoes, sorghum, so on and so forth, and looked at the different tissues, the panicles, the leaf and the root tissues at different developmental stages, took a range of rice varieties. Now, if you all know that rice can be categorized based on grain size into short grain, medium grain, and long grain varieties. These long grain the sweet smelling varieties are the basmatis probably, And the short grain varieties are a lot of short grain sticky rice varieties are also available. So we took a battery of these varieties based on the grain size, and then they categorize them in on the basis of their sensitivity or tolerance to various stresses. Some are tolerant to salt. There are some which are tolerant or moderately resistant to heat, being developed by various groups, and as tolerant to particular stresses. And tried to understand the response of salt stress, drought stress, high temperature stress on the plants. And also in this panel, we decided to include the virus infection. Now, these may look at different stresses, but aren't they regulated? Whenever temperatures are high, it results in increased evaporation, resulting in increasing in salinity on the soil. And if in case of rain-fed areas, they may even result in drought, which is a stress to the plant. And also high temperatures specifically increase or attract the insects to the plants. And most of these insects carry with them a battery of viruses. So we decided to keep a lot of these parameters, studying them individually first and then in combination, trying to understand how the plant responses are getting, just trying to understand how they are getting controlled in response to these single, double, or triple, or multiple stresses, to gain an understanding of the integrated regulation of the genetic machinery. And to this, the NGS or the Illumina sequencing came as a very efficient tool here, because we did a lot of small RNA analysis to mine and map the microRNAs, did a lot of transcript analysis to look at how the transcriptome is behaving, and also did a specific kind of sequencing called a degradome sequencing, which will help us identify the cleaved targets of these microRNAs. And then by integrating and analyzing this data, we moved forward. So what these profiles, a lot of libraries were sequenced, and what they threw at us were complicated graphs, or we can call profiles of these microRNAs and the transcripts, and these could be plotted in tissue-wise or stress physiology-wise or variety-wise. And if you look at these graphs, generically the overview, it doesn't throw us a unique pattern or a specific pattern here. They contain a lot of bars which are up and down regulated. So that means each data has to be looked into carefully and analyzed carefully. There is nothing stand out or a striking gene or a striking molecule which stands out and which says that yes, I am responsible for tissue development or stress response, so on and so forth. And also in this, just to mention that in the passing, that since a lot of small RNA libraries were sequenced, we could identify a lot of so-called novel molecules. Many of these have been validated, and there are still many which are being validated. And this is a huge data in respect to the Indian rice varieties which have been generated. A lot of it has been published, and because it's a live seminar, I will try to restrict my talk to the published data. And so what we could do by a series of, as a basic biology series of validation, was we could identify microRNA specific family members which were coming up as leaf preferential, root preferential, or flower preferential, and identified the corresponding targets which came up only in so like 397, the root target was not there, but in the leaf and the flower the target transcription was there, indicating how the targets are getting regulated. And based on these studies and using the help of computational softwares, a lot of intricate target microRNAs nodes were identified, and how these nodes were specific for their action in the leaf, in the roots, in the flowers, so on and so forth. Now, a lot of the NGS threw us a lot of data, so it had to be validated because there could be a lot of in silico noise there. And so the first step was to get into global validations. Specific microRNA arrays were made. Again, we have a lot of green, red mixed patterns, nothing clearly coming out, but we could identify expression zones in response to particular physiologies or in specific tissues. So for today's talk, I will focus specifically on one response to, so that it makes some sense to the listeners, and we'll focus on salt stress here. Now, when we talk about salt stress, we chose Pusa Basmati, the long grained, as the most salt susceptible variety. Then we have another variety called Pokali, which is very endemic to the southern region in India, and this is salt tolerant, and it can even be grown in seawater. So where we had two contrasting varieties here. This was a long grain susceptible variety, and this happened to be a short grain but a tolerant variety. Still, both are rice. The genome synteny is there. We expect a lot of sequence conservation. And as a control, what we took was we were fortunate to have this glycosyllase overexpressing rice, which was developed by Professor Sapori's group. And by overexpressing a biochemical pathway which involves two enzymes, GLY1 and GLY2, and these rice plants are transgenic rice plants are salt tolerant. Now, since this was overexpressed in the Pusa Basmati background, it gave us an excellent control. So we could now compare the susceptible, the tolerant, and the transgenic tolerant varieties. And in this panel, we also included another tolerant transgenic. This is a flockhouse virus B2 plant. Now, a flockhouse a virus is an insect infecting virus, and one of the protein coded by this virus B2 has been shown to impart tolerance to plants. This is now a different line of study. Papers have been published on that, but still we included this also as a transgenic, also as a control in our data set, just to understand how the microRNAs are behaving. So we took the panel of this so-called salt deregulated microRNAs and first started validating them by doing RT-PCRs and in different tissues and different varieties. And once we had this panel, we compared the Pokali and the Pusa Basmati. So this is one panel showing a comparison of the leaf tissues of those which are non-stressed and the stressed, and how the molecules are those molecules which are downregulated in Pusa Basmati, upregulated in Pokali, vice versa, so on and so forth. And we started looking at the graphs. Again, you see that there is a lot of haphazard pattern. But if we put a fold level, if we make a cutoff using say two-fold fold level, then we have specific molecules which are getting highly upregulated or downregulated in response in the tolerant varieties here, or in the susceptible variety. So if you look at these panels, these show, this is for Pusa Basmati, this shows that the fluctuation here is much more, both the upregulations and the downregulations, and this is what is seen in Pokali here. The fluctuations levels are comparatively much less, and we have very few molecules which are showing very high regulation. So there appears to be a tighter regulation of transcripts in the tolerant variety as compared to the susceptible varieties. Just an overview without getting into the details. So this was the kind of breakup that we have, that if we look at Pusa Basmati under salt stress and normal conditions, you have very few molecules which are getting downregulated. Sorry, you have a lot of molecules which are getting downregulated. The 0% stands here. A lot of molecules which are getting downregulated, and there are very few molecules which are upregulated. On the other hand, if you look at a similar comparison in Pokali, you have very few downregulations as compared to the upregulations. Now, don't bother too much about these colors, because this color is to show the fold changes from 0 to 2, 2 to 4, so on and so forth. But if you compare just the size of the graphs, then it is that there are larger number of down regulations in the susceptible varieties as compared to the tolerant varieties. So does this indicate anything, or is it because that they two of them belong to two different genetic rice backgrounds? To compare this, we looked at the glycylase transgenics. So this is comparing the glycylase normal plants with the normal Pusa basmati plants, or the glycylase salt stress plants with the salt stress Pusa basmati plants. The red dots are for glycylase and the black ones are for the Pusa basmati. Sorry, red dots are for the normal ones and the black dots are for the salt stressed ones. And if you see that there is again a haphazard pattern. So we took this data and we started looking at it in another fashion, where we compared the glycylase salt stress with respect to its own normal non-stress controls and Pusa basmati similarly. And here the red is Pusa basmati and the black ones are glycylase. Now, if you see, there is a lot of scattering of dots if you look at the red dots, whereas the gray dots are more or less more compact and they are more or less together. But the scattering pattern is more in compared to the gray dots. And this is what was reflected in the graph here. So you have again in the transgenics, you have a higher number of upregulations as compared to the down regulations, and but the fluctuations that you see is much less. And this is very much expected because now this is a transgenic where only one pathway has been overexpressed, although it is a salt tolerant variety, but we are comparing it here to a natural background Pokali, which can grow, which has naturally adapted itself to grow under saline conditions. But the microRNA profiles indicate that yes, there is if there is a change in the microRNA regulation, it is probably reflected in the transcripts, and these transcripts ultimately control the physiology of the plant. So, and just to answer your curiosity again on the B2 plants, again we have here. This is another pathway. This is a viral protein which is moving the plant towards tolerance. So we see a shift in the microRNA pattern here. So this gave us a lot of excitement that and also a validation that indeed the microRNA expression patterns are regulated in response to stress, and this will have a direct impact on the physiology or the growth pattern of the plant. So when we talk about microRNAs, we took those, we looked at them in one way, two way, multiple way manner to see which family member or which particular molecule is coming up in which physiology. All those kinds of studies were done. A lot of this work has been published also. So I'm not getting into details here, but what I want to stress is that microRNAs cannot be studied alone. You have to look at them with respect to the targets. And so we looked at the changing profiles of these microRNAs and tried to look at the targets. Now we had a wide range of targets which came up. There were transcription factors, there were enzymes, there were receptors, but we tried to focus for specificity's sake only on the transcription factors, how one master molecule regulates another master molecule. And this is to show that some of the microRNAs in these plants are regulating a wide range of transcription factors here, and most of these have there are reports of their association with one or the other abiotic stress response. So again, a lot of validations were done, and now we could focus on specific molecules, microRNAs, and their target transcripts, which the profiles, some of the profiles are shown here. And now this panel will not include only the transcription factors, but also some of the enzymes like helicases or some of the receptors. There were kinases and other enzymes. And this now turned out to be the core set of microRNAs, which were getting deregulated or which were getting intricately regulated in the tolerant plants vis-a-vis the susceptible ones. And three kinds of pathways came up. One was of course the transcription factors which act in regulating the genetic machinery. The other major pathway was the calcium signaling pathway, because calcium is an important signaling molecule here. And so and the third pathway interestingly happened to be the ROS pathway. And if we start looking at literature, we can actually stitch the three together, with the calcium signals influencing the ROS as well as the transcription factors to regulate the genetic machinery in the cell. So so overall we have some clue, but before making any claims, still a lot of scientific validations and proofs have to be done to understand what exactly is happening here. And this is and at the metabolite level, trying to understand what is happening. Then you can see the pathway in the red. So you have the glycolase pathway, you also have the glycolase pathway lighting up most of the protein synthesis and the respiratory machinery getting affected. So now if you can layer it, you can actually get a picture of how the signals are converging, whether they are calcium signals or ROS signals, they are converging on the transcription factors to regulate a variety of downstream pathways, majorly in the respiratory and the chain, and this would have an impact on the physiology of the plant. These are still maps. Most of this data still needs to be validated, but we have used a lot of computational algorithms to reach at this stage, because we need starting points to start our validations, and these are good leads to work further and to validate the work further. So based on this, we have constructed a kind of a hypothetical map to see how the microRNAs come up in specific tissues. Like in the root tissues, there is an increase of 398, which acts on the superoxide dismutase. This is to reduce the redox levels, or the others, specifically the other molecules coming up in the shoot or in the apical regions. So we have kind of categorized them with respect to the tissues, but they still need to be mapped on a developmental or a time scale before we can start reaching any conclusions. But this is a kind of a hypothetical map that we could derive at looking at trying to understand what is happening in Vocali, which is allowing the plant to grow happily in a saline environment, vis-a-vis another plant like Pusa basmati, which gets heavily affected by salinity stress. So some of the papers to show, but many of them have been published. Now, likewise, the work has been done in response to high temperature stress. I am not going to get into the details of the work done here, but a lot of validations and a lot of time kinetics were done with specific miRNA molecules. And this led us now to recognize common microRNA nodes which were coming up in response to high temperature and salt stress. These are the common nodes. These nodes would operate in response to both high temperature stress and salt stress. Coming alone to a particular stress will be very different. And if you look at the targets that they are regulating, there are the ROS enzymes, there are energy converters, there are hormone regulators here, and different those which regulate flowering. And this is important because when we have a plant growing in stress, what we see is a reduction in flowering, and this this is an important lead which can be followed further to understand how these common nodes are differentially regulated in response to different environmental stresses, and what is their impact in response to the plant. And if you remember in the first slide, one of the first beginning slides, I had mentioned that temperature has a differential response on the growth of the plant. While there is the nighttime increase in temperature seems to be more detrimental as compared to a daytime increase in temperature. So what is it? Is that the presence presence or absence of light has got something to do with it. And we have important leads now. One of the manuscripts is under communication, which shows that light is actually playing a role there, because light is constantly there. Plants cannot survive without light. And some of the light signals also overlap with the stress signals to influence the physiology of the plant. And our focus, of course, is to look at how the microRNAs are getting deregulated, to understand if we can understand even one of these nodes, the functioning of or the regulation of one of these nodes in response to the different stresses, it will be a lot of knowledge gained. So again, based on our studies on heat stress, on salt stress, and different developmental stages, we have built hypothetical maps of how particular microRNAs can be coming up or going down with different stages, and to affect the plant development. And a lot of this data is still under validation. Now, since a lot of data has been generated and our hands are amply full, we decided to put or compile this data in the form of a web server. This is freely available from the ICGEB website, and this is called ARMOR, for a RISE microRNA mRNA interaction resource. It carries a lot of information on the different microRNAs on their associated transcripts. It carries information on different microRNAs from 7 Indian plant varieties, and is freely available to use. You can pick and choose what it can be applied for. What we have done is we have used some of this data. I'll just tell you how we have branched from here. We have used some of this data to understand the sugar accumulation using sorghum and sugarcane as the crops, because sugar accumulation is affected both in sorghum and sugarcane. They are closely related cereals and provide an excellent system. Again, a lot of mapping of microRNAs and targets is underway, and we are trying to understand how it is affecting, because sugar is the source of energy, whether it is in the form of seed, in cereals, or in the sugar content of sugarcane, and is heavily influenced by salt. So I purposefully included one of the slide prepared by Neha, my student, and she has done some lot of good work on microRNAs. And while we were having this panel of microRNAs, and just as an explanation, we can categorize them into 2 kinds of molecules: those which are conserved between plants, and those which are non-conserved or unique to specific species. So like for rice also, we have specific MIRs which are unique to specific species. And these unique molecules are normally targeting the enzymes and the receptors generally. And whereas the conserved molecules conserved between different plants are the ones which are targeting the transcription factors. So while a lot of interest is there on understanding this particular interaction between the conserved microRNAs and the downstream transcription factors, because this will impact hundreds of genes, we also had some specific interest to look at the unique microRNAs and how they are influencing what role they are playing in influencing the physiology of the plant. So one of these microRNAs were picked up and found to be very specific to rice. Interestingly, it was seen that it showed even differences in the genomic locations when we started look between the Japonica and the Indica rices. There were larger number of alleles in the Japonica varieties as compared to the Indica varieties. So initially we thought that it has probably got something to do with genome sequencing. Either the chromosomes have not been fully mapped, that could be the reason, or the sequences still need to be understood. But then we did a parallel study using a lot of different rice varieties. This was a collaborative initiative with an Australian group, and what we found that indeed the number of genes of this microRNA varies across rice genomes, and and we have leads to show that it impacts the grain physiology and the stress response of these rice varieties. The lot of this data is still unpublished, so I will not get into the details on it. But what we did was we started raising some transgenic with these with this particular microRNA, and to show you the slides, you can see the difference in the plants. You can see these plants are much more denser as compared to these ones, and the ones in the arrow are the microRNA transgenics as compared to the wild type plants. And just to show you a picture of one of the maturing plants, so this is what a mature or transgenic plant would look like as compared to a normal plant. It's more robust, gives us more seed. So we are still trying to understand the mechanisms behind this, but then this is just to show that a lot of this research has helped us identify a microRNA which has the potential to boost yield of rice plants. This manuscript is also under publication, so I will not spend too much time on the slide now. And also we have been talking for a long time, so I should end not without thanking my team members, those who are there in the lab presently, and for those who have been associated with my group, because it is because of them that I am speaking with you here. And of course, I need to also acknowledge the funding agencies and my mentors who have helped me in this journey. Thank you so much. So I would be happy to take questions if any.
Hello, ma'am.
Yeah.
We collect few questions for you.
Yeah.
You can find in the chat box. There is a chat option just beside the green screen.
Okay, maybe I have to stop sharing my
Yes, no problem. You can stop.
Where is the chat box?
Just beside the blue screen. Yeah, where you start the screen share option.
No,
there is a chat.
Yeah, I have chat box. Yes, okay.
So there is a name. The name of the participants is within the brackets, and the questions is followed.
There is one by Kritika Sharma. How can we confirm which particular microRNA regulates the expression of a particular gene? Okay, so microRNA action is highly sequence dependent. So from the sequence of the microRNA, there will be a reverse complementary site in the target. And based on this, several easy computational scripts or algorithms can be designed. Even manual searches can be done if you are using only one microRNA at a time, but that will be a little too tedious. So a lot of computational tools are available which can help you identify the targets of these microRNAs. Another question is by Dinesh, which says, can we use rice microRNA data in sugarcane for drought stress? And is there any problem of genome complexity on microRNA study? Okay, so a lot of synteny is there between the grass family. And since microRNAs are coded by the genomes, it is expected that at least the conserved microRNAs will be there between the different plant species. There may be slight sequence variation depending upon the genome. There could be SNPs, or there could be variations in the precursor structures. There could be variations in the promoters as well, which will have implications in their expression patterns. But over and above that, a straightforward answer to the question is yes, you can use these sequences to identify molecules in your data sets. But lot of validations, experimental validations would have to be done to confirm that they actually exist. And also on the nature of study cannot be directly replicated. You will have to design your experiments carefully to answer the questions that you have raised. I hope I'm able to answer this question. So I'll take another one. How can we use microRNA data of virus resistance in the development of plants' resistance to mycoplasma? This cannot be a straightforward answer. RNAi per se is the first line of defense for resistance to any kind of pathogen. There would be involvement of siRNAs, microRNAs, tacrRNAs, and other molecules which will act in together to regulate the resistance versus the susceptibility response. Also, we need to take keep in mind here the R genes or which forms the other the R AVR genes which are also involved. So this is a complex. This is a full subject in itself. A lot of study has to be done. But yes, what I can say is microRNAs are deregulated in response to virus infection, and some of these deregulations are directly responsible for the phenotypic abnormalities that are observed in the leaves, in the flowers, in the seed, so on and so forth. Other than that, there can be virus encoded microRNAs which will help the virus in invading the host. There could be a host encoded microRNAs which can be acting in favor of the plants to resist the virus infection. There could be some of the host microRNAs which could be helping the virus. There are many things in this, so a thorough knowledge of the literature would actually be required before I can fully answer this question. Okay. Coming to the next one. Can we use this technology for direct yield improvement? Of course, yes. One example I have shown. The acceptability of these plants, of course, is like any acceptability of any other transgenic plant, because this is transgenic, because it comes with the antibiotic selection markers, it comes with foreign promoters. But yes, this technology can be used for yield improvements. The other line is what we are trying to work on is the identification of microRNA markers which can be used for breeding approach. That can be another approach, but then these markers, a lot of thorough study has to be done in identification and acceptability again for these markers, but the potential is definitely there. All right, so Jyotsna has, what are the other ways to validate miRNA except qPCR? So other than, I will not say qPCR, it's a generic term. Other than PCR-based technologies, there are different PCR-based technologies. You can do a direct Nathan, you can do a Nathan plotting, there can be cloning and sequence validation. These are the classical methods that are available. Is there any miRNA modification that has been reported to have negative impact in plants? miRNA modifications have been reported. miRNAs do undergo post-processing, I will call post-processing modifications, because miRNAs are processed from their transcripts. There can be addition of bases at the ends, either 5' or 3'. There can be deletions of bases at the ends, and you will understand that since the first 10 or the first 12 nucleotides are very important in target recognition, so any alteration in the 5' terminus will have a direct impact on the target. But it will be too early to say whether they have any negative impact in plants, because the understanding of these modulations and the stress-induced modulations of these microRNAs and the functionality still needs to be understood before categorizing the impact as a negative or a positive impact. It will definitely have an impact, but it will be too preliminary to categorize that impact as a negative or a positive impact. So I'll move on to the next question, which says, do you know of any miRNA which are present only in abiotic stress tolerant and not in sensitive genotypes? Yes. there are definitely microRNAs which are I will just reword your statement, which are present in the tolerant species and whose expression is either highly reduced or they are absent from the sensitive genotypes, and vice versa. These changes do happen. They may vanish, or they may, or I shouldn't say vanish, they may disappear from one tissue, appear in another tissue. They can be overexpressed in a particular tissue. Why I don't want to use this term present and non-present, because there are limitations of our detection techniques. We cannot find a particular microRNA in a particular tissue, probably because it is present in quantities which are too less to be detected. Its value may not be zero. It can be zero, but it need not necessarily be zero. So we should be very careful, because we have limitations unless and until we totally rule that out. But yes, there are drastic changes in the expression of microRNAs, which can give us an appearance that it is coming up in a specific genotype and it is going away in another genotype, and its relation to tolerance and sensitivity to a particular stress has been studied. So there are a lot of reports on that.
Okay, ma'am. Ma'am, there is it's okay. You have answered many questions, and more are like similar and perform your talk. So we are not taking these questions, but we send all the questions to your mail after the webinar. Now I request Shoma, please do a short interview.
All right. We are now moving ahead to the interview section of today's webinar. Today is our 20th webinar of the series, and we are honored to have Dr. Mishra here with us today. Ma'am, my first question to you is that you are a very successful plant scientist, and we would love to know how you built your career. Could you please tell us about your journey so far as a plant scientist?
I am a plant scientist. I would not like to use the word successful here, because there are different parameters of judging success, and I really criticize myself. So I will not use that term successful. But yes, plant scientist, because of my interest and for my love in plants. As a child, I was always, as a teenager, I was always interested in plants, home gardening and all. That time, the thought never crossed my mind that I would end up taking science as a career. But then it attracted me towards subjects related to botany. They caught my attention. Progressed further, had this curiosity of learning more and more. And this kind of shaped my journey without my actually realizing this thing. But now when I look back, I understand that it was my curiosity which kept me moving forward in that direction. And I did try to switch fields. I did try to jump out of the plant field and try my hands, thinking that molecular biology is the same. But then I came back. I understood that my interest lies in plants, and this is why I am here. So and the curiosity still keeps me moving, because I'm still interested. I am just asking basic questions. What happens and why does it happen? And this is what is driving me throughout.
Wow. Ma'am, my next question is that along with food security, what are your thoughts on nutritional security?
Absolutely a requirement of the day, because when we talk, I think when we talk about hunger, it is not only having your plates full or having the resources to keep your plates full. It is also having a complete nutrition. So nutritional security definitely is the need of the hour. And there are dedicated groups who are now working towards enhancing the nutritional content of plants, whether it's iron or whether it is making it gluten free, or different aspects of it. But ultimately, see, it's actually a balance. You have to eat, you have to eat to stay healthy, because that's a form of medicine. And then you have to be nutritionally sufficient to keep your immunity and your health boosted. So although food has to be, primary thing is food has to be there, your plate has to be full. The second thing is it has to be nutritionally enriched, so that what we are eating gives us the required nutrition. So it's you cannot take both the things in a standalone manner. They both have to go hand in hand. You can't have less resources which are nutritionally enriched so that only a few can, or you have only half a plate full. So you need a plate full as well as you need it nutritionally balanced. This is what I would say.
According to you, which sectors of plant science research will prosper in the near future?
All aspects. All aspects, because see, in science, if you don't have basic knowledge, you cannot move forward. There has been a thrust, there has been a focus on translating knowledge, which is very good. We need to be innovative. Even RNAi, if you look at it, discovery began early 2000. 20 years down the line, which is not too much of a time considering the scientific time scales. You're already translating the knowledge either in medicine or getting crops or making virus resistant, so on and so forth. But we still lack the knowledge. So what we have done is we have discovered a tool, we have started using the tool, but to realize the full potential of the tool, we need to understand more, we still need to investigate more. So all aspects of science, whatever more we can discover, whatever more knowledge we will discover, would help us move forward in translating. So that is the key. So there cannot be a line drawn which says that this is more important than the other. The complete understanding the complete plant biology, every aspect of it, and you will not know where you can apply that knowledge to reap the benefits.
So true. Ma'am, the last question. What would your words of advice be to the new research fellows?
I would say, whatever you do it with sincerity. Don't just get into science because you are all fascinated or momentarily attracted by the scientist, especially in this COVID era, virus science, everything is, these are the key words which are happening there. Plant biology, plant science is equally important. If you have medicines and you don't have food, that it's going to be a big problem. So choose wisely. Don't just get into an area because that is in news or in media these days. Whatever you want to do it with full sincerity. This is my advice. And also, you don't think that you'll step into a lab today and step out with a product tomorrow. That doesn't happen. Everything requires a lot of patience, lot of hard work and sincerity. If you can work with the combination of this, you will definitely succeed in whatever you are doing. That includes science. So since I belong to the science field, I always ask my people, my group members, think, work and works sincerely. That's it.
Great words of advice from you, ma'am. And it was a beautiful presentation. I'm sure all of the viewers liked it very much. And we've also got many positive feedbacks. Okay, so let's go back to Shubha now.
Thank you, Shoma. Thank you, ma'am, for accepting our invitation. It is a very new platform, and we are trying to build it because our intention is to promote plant science, and obviously it's freely and for everybody, so they can ask. And very interestingly, few questions we have found that for today's webinar, someone asked that, can I do research work on your lab? So few questions are there. I want to study microRNA in my plant species in your lab. So these are the things which we are feel that when we are MSc or we are in the pre-PhD, if we know that scientists, those are available in our reach in India or others, or we by this platform we can able to learn so many fellowships are there, like how to get into ED or how to get into other national and international institutes. So we think it's fruitful for us that we try to build, and obviously we love the interview sessions, because we can't find these things in your website or any other than your autobiography. So so it's a honor to us to have you, and I also thanks to Neha give me the idea that we can ask Neeti ma'am. So it's a wonderful to have you, and I'm sure in future, because
No, I think I need to thank you and many congratulations for starting this initiative of drawing attention of the young minds towards plant science, because that itself is a big thing. Other than doing the science, attracting them, catching them, telling them exactly what is happening, because that is now, I think, the need of the hour. From virus, human epidemics, we are moving towards the hunger epidemic. So this is really the need of the hour, and thank you for including finding my lab work and my work important enough to be discussed on this session. So thank you very much for that.
Thank you, ma'am. And
Thank you
I
so
request,
much, ma'am.
yes, I request Neha. Thanks once again to have ma'am for today's.
My pleasure, Shiva. Thank you.
I also thanks to Jacinta, our team member. Ma'am, this session is over now. I collect all the data and the rest of the questions and some appreciations of your talk, and I will send you a mail with the, all feedback, all the nations-wise outcomes and everything. And I send you a mail by tonight or obviously by tomorrow.
Thank you so much. Yeah.
Thank you.
Nice interacting with you. Bye.
Okay. Thank you. Bye. Now I request my team member thanks. We successfully complete our 20th webinar. Now I'm request Soma, can you go for the website and little bit for 5 to 6 minutes, can you explain where the certificate can download?
Sure. Shall I share the screen?
Share your screens and go for that. And Jacinta and Neha, you can we meet.
No,
and
I would like to stay and listen.
Okay. It's great. So we are still live. I'm answering and collecting few questions from the YouTube. Neha, you can do that. Jacinta, hi. Jacinta also there. So
it's late for her.
Yes, it's okay. So you start the sharing, and I request you to update all the guys that where the they can find the certificate, where they should collect the certificate, where they going to ask and suggestion for new topic for the webinar and for the discussion, which are our upcoming webinars, and that's it. Okay.
All right. So this is our temporary website. We have a new one coming very soon. That is bioengine.com. So
can you full screen your screen, because it's not full screen.
Okay.
Is a option there is a 3 option at the top of the right side cross. There is a 4 square and one is a single. Where you open the new tab, there is an option in between the minus and in cross. Okay, let's see. No problem. Okay.
It is full screen actually. I mean, I don't know what you are.
Yes, for my view, it's not.
I'll ask Neha. Is it full screen?
Okay, it's okay. It's not look like not, but okay. I understand why it is.
Yeah, it's full screen.
Okay.
Yeah, it's full screen.
All right. So this is our temporary website. We're using Google Sites for this, and you will see that there's a notice section, and then we have the upcoming live webinars. So the next webinar is on 19th October, which is from Dr. Dhananjay Gotarkar from ERI. And then you can just visit the website and you will see all the upcoming webinars. And then you scroll down, and here is a new section that was not previously there. This is the upcoming live discussion that we were talking about right in the beginning of the webinar session today. So we are starting this live discussion session. It is a more peer-to-peer discussion. Students, mainly scholars, mainly PhD scholars and postdocs who have some sort of experience in research or different aspects of research. So you can see that there are 3 live sessions that have been posted here. The date and time is there. So our first Bioengine live discussion session is on 17th October at 12 noon for IST. And there's the world clock. You can check what time it will be at your place. And also just below that, there's a registration link. Please click on the link and you will see a form. And shall I click on the link, Shubho? Okay, let's see what happens when you click on the link. See, here is the form. When you click on the link, you get the form. This is for 17th October. The topic is credit, a rapidly adapted system by international journals to determine the role of the authors and their area of contribution in a manuscript. So I hope if you have seen the recent journals, you will find that they are using a new way to acknowledge the contribution or to determine the contribution of the authors in a journal. So this is a new process. It's working for quite some time now. So this discussion will be regarding that. And you can participate here by putting in your email address, your name, your profession, your last degree achieved, your institute name, your institute address, your country, and you can put suggestions of any topics that you want to be discussed in the future. Okay, and you can submit this form. Okay, so let's go back to the website. And if you scroll down, you can see that there's a place where it can be a speaker, writer for webinar, discussion, and blog. So we are accepting requests. If you want to be a speaker on our platform, if you want to write a blog on our platform that will be posted on our platform, if you want to be a part of these discussions, also there's another link right beside it that says topic suggestions for a webinar discussion. So you can put in your suggestions for a topic. If you want to, like, if there is a topic that has been bothering you which is related to research, definitely research, then you can put in your suggestions here. Also, I would like to add that these Bioengine live discussion sessions will not be based on plant science research alone. We are accepting discussions requests, and we will be talking about research in general and mainly of life science, because life science research is unique. I mean, it requires some sort of wet lab. So I mean, for Bioengine live discussions, our main focus is to address the issues that researchers or new to the field of research, or who are thinking about going into research, are facing for the field of life sciences. Okay, so if you are any field of life science, you can please join in the discussion sessions. And if you scroll further down, you will see that there's a live webinar feedback. In the brackets, it's a certificate application. So here are the lists of the all the previous webinars, and the hyperlinks when you click on these hyperlinks, then you will see that the Excel sheet opens, which has the names of all the applicants who have applied for the certificates, and there they have been allotted a file number. And when you go to the Google Drive by clicking on the certificate link right beside it, it will go straight to the shared Google Drive where you can download that file number, that particular file number that is your certificate. You just download it from there, and you have to download it. We are not sending it by email or anything. You all just get into our website and download your certificate after 2 days. We need 2 days to make the certificates. Okay, so that's all. And at the very bottom of the page, you'll find there's a submit your testimonial. The link for the testimonial is also there in the tabs, right in the tabs on the top of the website. There are the about webinars, interviews, blogs, posters, and people, and then the final one is testimonials. So we are collecting, as I told you that very soon we will have our new website, which will be www.bioengine.com, and we are collecting testimonials for that website. So you can put in your picture and your testimonial, and we will post it in our testimonial section in our website. So that's all, I think, for the website. And also there are other tabs which you can go in and have a look. There are the posters. We are collecting posters. If you have a pre-published poster that you really want to show everybody that, okay, I would like to show my poster here, I really like this poster, you can please send it to us and we will post it here. If we find it good, we'll post it here. And there's a page for blog. You can write in popular, we are accepting popular articles related to the field of plant sciences. And here's the interviews tab. You can see the interviews that have been posted in our YouTube channel. There's a direct link. You can just get into our website, and if you want to watch a particular interview that has happened in the past in our webinar, you can just click from here, and there you will have your interview video open. Okay, so let's go back to home page. All right, so I'll just quickly say what webinars are coming up. So we have, I just said that our next webinar is on 19th October. The time is 11 a.m. India Standard Time. And the topic is molecular and physiological mechanisms of drought tolerance in rice, a tale of backbencher gene. So this this webinar topic, the speaker is Dr. Dhananjay Gotarkar from Erie, Philippines. And the next webinar is on 3rd November. So no more webinars in October, then we straight go to November. On the 3rd of November at 10 a.m. Please note the time. It's 10 a.m. India Standard Time. And please have a look at the world clock for the time at your place. Okay, so the speaker, she has been on our platform and she has given a wonderful lecture on soft skills. So her name is Dr. Konkona Khoshal. She will be talking on soft skills, create a positive and productive work environment. She will be speaking from Los Angeles, USA. And the next webinar in the month of November is on 6th November 2020 by Professor Ashwini Parikh. He is from the Jawaharlal Nehru University, India. He will be speaking on the topic towards raising crops for saline and dry lands. The time is, as usual, 11 a.m. Okay, so the next webinar is on 24th November. I mean, next to 6th, after 6th November, it is not 24th November. By Dr. Ricardo Oliva. He will be speaking from Erie, Philippines. And the topic is from breeding to genome editing, long-term strategies to contain plant diseases. And the timing is 11 a.m. And after that, there is a talk on 4th December. So after November's talks, the December talks are there. 4th December, the talk is by Dr. Manish K. Pandey. He will be talking from ACRISAT, India. His topic is transnational genomics for achieving higher genetic gains in groundnut. So this is our first groundnut related talk. And kindly note the time because it is different. since ICRISAT is working collaboratively with Africa a lot, and a lot of research is being done in Africa, wonderful work. So for the people of Africa, and there have been requests that please the timing could be more suitable for us to view. So the talk timing is at 2 p.m., which is quite later in the day for us, and I think it will be a good time in the morning for African countries. So please, all the people from Africa, you can watch it, and you don't have to worry about being very early in the morning. Okay, so the topic, and we are trying to organize many more, talks in December. So I think that's about it. What do you say, Shubham?
Yes, it's okay.
So I'm stopping my screen sharing.
Okay. Thanks a lot for everyone to attending to our this webinar. We expect all viewers for our next webinar. And as Shoma says, and as the starting Jacinta says, that we start a discussion topic. It's more focused on the students and scholars. We will discuss which are the very basic but very important for our researcher life. So it's not a particularly plant science or talk, but we can discuss the instrumentation, methodology, or we can discuss the how to write, how to publish, how to find a good journal, how to keep in the mind when you're writing something, these things. And for that discussion, I request all of you, and I already request many of our viewers, that if you want to participate in the discussion, if you want to be a panelist and you have something to discuss, it's not like just you want to a panelist. If you have something to discuss and important for others, you can text us, mail us, or you can put your request through the website. So we can go through your profile, and we may keep you one of the panelists on the Zoom to discuss, which is more live and interactive. So between the discussion, the questions and answer will definitely help the YouTube listener. But we collectively like questions from the YouTube and try to discuss. But this discussion should be within an hour. So it's 1 hour.
Yeah, if we start talking and then it goes on, because we love to discuss and talk about research. So we'll keep it short for everybody's sake. And also you could share that we have been having these type of requests all along, like when we were having webinars from so many students.
Yes,
They want
that is the one
it's
of
on
the
demand, actually,
yes,
on popular demand.
because I got many mail and many messages that we need this type of things. That's why I think, and okay, we can arrange this type of, because I personally also feel that when I start the writing, I don't know the actual concept of the plagiarism, what the mean is plagiarism, and I have many questions. If I copy paste something from my previous paper to now, what happened? I already mentioned in my thesis, now I writing it in my paper, what happened? So I am interested to discuss such a thing, which I sorted out by by uh, by my own sometime, and sometime by getting the advice from a seniors. But if you find something like that from a discuss group in a national or international way, and it's free, so we can, if we try to collaborate, we can collaborate, and I'm sure that it's benefit many of us. And if we can able to join due to time problem, due to because we have so free.
Sometimes, people who are new to research, they need somebody to talk to because
Yes.
they get,
So
sometimes they cannot
they can
access
also
people.
understand. They can also understand that okay, it a process. So they can prepare themselves very beginning, not at the end when you're ready to write your thesis and you know that this happen. So it's very difficult. Like one is a plagiarism, another is how to remove the plagiarism by the softwares, and most of the softwares are famous. Like Turnitin is a famous plagiarism removal software, but it's paid. You need subscription for that.
What do you
So
mean by removal of plagiarism? I mean, isn't it like identifying plagiarism?
Yes, identifying plagiarism, matching the why it is shows the plagiarism, these things. So we can't. I don't want it to discuss here because
All right.
we have a discussion session for
Definitely.
there, and we
Looking
try to,
forward.
yes, and we try to include few people. Those are the either academic writer or those are the known this process. But here I try to explain that the tax and free softwares which can use to remove the plagiarism by the free softwares, and we can live demonstration also these softwares if possible, if time is a permit in during the discussion session. So it's one of the aspect is the plagiarism. Like there is a management of your citations. So there is a citation managers are there. So these are the small things where you can ask those are the expert or those are known to it. And for the first discussion session, I will start it because I decided to launch it, so I have to start it for the first time. But I already gathered 5 discussion with the speakers, and very slowly I put their information in our website one by one, because this discussion is one in a month is a frequency of that. We not because we also have to organize the webinar. So
Webinars,
it's
yes.
very difficult for me when I work my lab work and also through bioengine work. So that is the reason that I keep the some balance to my own work and this work. So we try to in a month, one in a month. So let's see what's happened to next. So we gathering in the discussion, that is the 17. Then
Right.
we come to the 19.
Or next time we meet, it will be on 17th, right?
Yes,
Right.
17,
Okay.
12 o'clock.
12, right. The timing is 12 noon.
No.
All right. It was lovely
Okay,
to be a
thank
part
you.
of this webinar. See you.
Thanks, Shoma, and thanks all the participants. Someone asks feedback link till, but it's okay. I'm giving the for once more, but this link is deactivated in 1:30. Okay.
Okay.